Hello everyone, welcome back to another session of RNA biology. So we were exploring the different mechanisms and the way in which X chromosome gets inactivated to maintain the dosage compensation. And in nutshell, we know that the X-ist means X inactivation specific transcript, the non-coding RNA is accelerating the chromatin compaction or the DNA compaction. So it will facilitate those mechanisms that will prevent the loosening of the chromatin and accelerating the tightening of the chromatin. So if you have acetylation, chromatin becomes loosened. If you facilitate deacetylation, chromatin becomes tightened. And if you facilitate methylation, that will become a tight chromatin. So this is the main ways through which the X-ist is allowing the compaction or the condensation of the one of the two X chromosomes. So let us see what are the main features of inactive X chromosome or inactive X territory. First feature is X-ist RNA itself. It's a long functional RNA that initiates the silencing in cis. So cis and trans word we have used it. So easiest way of understanding cis is within the same chromosome, within the same gene, within the same vicinity. If it is in a distant place, that is called trans. If you and your friend sitting in one room, you are talking to your friend while sitting in the room, it is called cis. And if you are talking to someone over phone who is in another country or another state, that is called trans, in easier way of making you understand. So X-ist acts in cis, that is on the same chromosome where it is being produced, and it acts on the same chromosome. Then comes the polycomb group proteins. They are called PCG proteins. What are they? They are mainly of two types, PRC2 and PRC1. PRC2 is polycomb repressor complex 2. PRC1 is polycomb repressor complex 1. So PRC2 contains proteins such as EED, EZH2, SUS12, etc. Many components are there, and EZH2 being the catalytic component, catalytic molecule. Same way PRC1 also have got plenty of molecules, BMI1, PHC1, PHC2, CBX2, CBX7, ring 1A, ring 1B, like that, so on and so forth. So these proteins congregate together onto the chromatin, and it can facilitate specific modifications on the histone proteins, because of which you can create a repressive chromatin. That means chromatin that is not available for transcription. That is the meaning of repression. What are the modifications that takes place? Histone 3 lysine 27 trimethylation, mainly done by PRC2 complex, and histone 2A lysine 119 ubiquitination, mainly done by PRC1, and histone 4 lysine 20 monomethylation, histone 3 lysine 9 dimethylation, histone 4 hypoacetylation. So these are all some of the related changes that takes place in the histone level. Histone is part of this chromatin. So without histone there is no chromatin, and the modification, chemical modification of histone decides whether or not that chromatin should be tight or loose. So this is decided by the modification. If histone is tightly bound to the DNA, then it is a solid, tightly packed chromatin, and we call it as heterochromatin. If it is loose, it is loosely connected to the DNA. So enzymes like DNA polymerase enzymes can come and act onto them effectively, and we call them as a euchromatin. So histone variants macro H2A is specifically seen in the bar body or this inactive X chromosome, and histone 1 is another feature that is seen associated with inactive X chromosome. And there are other bunch of proteins such as SAFA, and that is a nuclear matrix associated protein, and also BRCA1, BRCA1, and that is also seen to be associated with this inactive X chromosome. And also it exhibits some features, some uniqueness. What are they? Exclusion of RNA polymerase 2. We know what RNA polymerase 2 does to a DNA, that will facilitate the expression of mRNAs. So it is not welcome to this inactive X chromosome. If RNA polymerase 2 comes in, there can be some expression that can sabotage the dosage compensation. And then comes late replication. In a cell division, we know in S phase, normally cell have got multiple phase, that is G1 phase, S phase, G2 phase and M phase. M phase is for mitosis, G1 is the growth 1 phase and S phase is synthetic phase and G2 is the second growth phase and then goes into M phase. So normally the DNA replication takes place in the S phase. If S phase all the DNA are replicated, then it will get the green signal to move on to the G2 phase. However, the inactive X chromosome replicates very late, just like a student coming late to the class. So he will miss many of the features, many of the qualities or many of the instructions given in the class. Like that, this X chromosome will replicate very late. And you can guess what could be the reason, because it is so tightly compact, it takes ages to unpack it. It cannot be done easily. So it takes lot of time to unpack it, and this makes the replication very late, and it comes with a price. The system cannot monitor whether is there any mutation, is there any trouble, etc., because there is no monitoring is happening. It is a discarded or neglected X chromosome, and this is okay to some extent because it is not expressing. But however, if there is a chance by mistake, if some leaky expression comes, that can be quite dangerous for that organism, because that will accumulate lots of mutations usually, because it is not been repaired, because it replicates very late, and once it is just finished replication, there is no quality control machinery can act on it. So this is what you should keep in mind, and we will see some examples whether what is the implication of this late replication has onto the mutation rates in the X chromosome. And exclusion of H2A BBD. So this is an isoform of histone 2A that will be excluded. So these are all the three features associated with X chromosome. And we can see some examples of the imprinting. What is imprinting? Imprinting basically means something is been told to you straightforward. You are supposed to do this. Like many such examples are there. Like if I give some examples of human behavior in imprinting, it will be much easy for you. What is imprinting? If you are supposed to behave so and so, like if you are going to school, you are supposed to wear uniform. If you are going to for a function, you are supposed to wear decent dress. So these are all something information which you get it from your parents. You will not go like a household dress to attend a function. So certain behavior in human is imprinted for no serious reason. If you go to a function, if you go to a big grand function just by wearing a shirt and pant, nothing wrong with that. It's a civilized dress because it is civilized dress in the office. So why not in a function? Why we call it as a party wear or a function wear, etc. Why such a different category should be there? It is imprinted in your mind. Oh, if I am going for, and you may apply some perfume, maybe you will put some makeup, etc. Usually you don't do that when you are going to your office or school or something. You will go just like a routine person. So this is one example I can tell you, imprinting. So genes are also like that. Some genes, they say, okay, this particular gene should not be expressed. Why? Because this is coming from paternal copy or father's copy. Or this gene should not be expressed because it is coming from a maternal copy. Or this should be expressed. Why? Because it is coming from a father's copy. So like this indication or this instruction given can be easily called as imprinting. So let us see how inactivation takes place in a imprinted. One imprinting we can say, imprinting mechanism we can say, in a female, if two X chromosomes are there, one must be made inactive. This is an imprinted behavior because it has to be, it's a rule, cannot be violated. So you can see in placental mammals and marsupial mammals, marsupial mammal means it will deliver the baby at a very small size and shape. The development doesn't happen inside the womb very effectively, and the small baby will grow inside a pouch. Mother's belly will have a pouch and it grow inside. All of you have seen kangaroos. Whenever you see a picture of a kangaroo, naturally it will have a small baby. Of course, only for female such feature is the small baby inside the belly pouch of the mother. So it keeps growing inside to a significant extent. So they are the feature. They have a marsupial pouch. So that's why we call it as a marsupial mammal. Examples we know: kangaroo, koala, Tasmanian devil. So these are some animals which follow a unique set of X-inactivation, which by default the paternal copy is inactivated. Whereas in placental mammal, it is not like that. Like mouse, cat, humans, etc. Initially, it is the paternal copy that is inactivated. Later, during the developmental stage, there is a selection taking place. Selection in the sense a randomness is coming into picture. It can be either maternal or paternal copy can be made inactive. Whereas marsupials, they follow a steady state pattern of paternal X chromosome inactivation. So it's an example of a cat, and this cat is called calico cat, and here you have an example of Tasmanian devil, which is a marsupial carnivore. That is only one member in the marsupial group which is a carnivore. Rest of them are mainly herbivore or omnivore. This is a strict carnivore, means eating meat. So calico cat is something interesting. Like, we will go into the details of why calico cat is looking like that. We call a calico cat so if it has both yellow and black color together. You may have seen cats with white color and yellow color, or white color and black color, or white alone, or black alone, or yellow alone. The moment you see a cat that has got black and yellow together, white may or may not be there. White is optional because white is not a color, by the way. Black is a color, yellow is a color. If you see a cat that has got black and yellow together, don't need to look what gender it is, because that must be a female. You don't have to bother. No male cat will have both this color, yellow and black together. We will see why. All we can imagine is it has something to do with X chromosome, because X chromosome is randomly inactivated. Male doesn't have because females have all the chromosome in 2 pairs. Males also have all the chromosome in 2 pairs except a sex chromosome, where males have only 1 X, females have got 2 X. So that holds the clue, and the coat color is linked to the X chromosome. So I'm not saying that 1 X chromosome always will have yellow or 1 X chromosome always will have black. No, not like that. There can be both the chromosome black or both the chromosome yellow. So that time you can't distinguish. But if a cat has both this color, that means the X chromosome is randomly made inactive on the skin. So we will see the genetics behind it. So random X inactivation in calico cat, how does it work? In male, it has got XY and XY. So here you can see XY is written orange in color, and here XY 1X is shown in black in color. Y is given common ash color. Whereas in female, you can see 2 versions, in fact 3 versions. One is 2X black in color, 2X orange in color, and 1X orange, 1X black. We should understand the orange color is dominant when it comes to skin color. Orange color is dominant over the black one in the case of calico cat. So what we should understand, if it is in genetics, you would have studied dominance and recessive. You may have studied the Grigor-Mendel's experiment on Pisum sativum. If you cross tall pea plant and dwarf pea plant in the F1 next generation, all of them will be tall. There is no intermediate size. There is no representation of the short at all. But in the next generation F2, if you self them, you will end up getting 1:3:1, or depending upon whether it's a monohybrid, dihybrid, accordingly you will have a variation. So what we should understand here, X and Y is there, we know that Y has no color. So naturally this animal will be orange in color. Orange means yellowish orange. If male X and Y here, the black one which is recessive, not dominant. So here in the Mendel's experiment, the tall color is dominant, short or dwarf one is recessive. So dominant means it will show full-fledged one character, whereas short one has to have both the loci. In a diploid organism it will have two locus. Both the locus it should have the characters governing the shortness, then only it will exhibit. So we usually refer to this as homozygous recessive. So think about this male, if it has got orange, that will be expressed, and naturally this one can assume it is dominant. In male, if it has got one X that is having black color, the black will be expressed. It is recessive but there is no orange, so naturally it will be expressed in black color. And you can also assume no male can have both of them together because it is in the X chromosome. How can a male will have both X chromosome together? Then that male has to be XXY. That is not possible usually. In female, you have both the X chromosome, which is meaning for black, then the organism will look black in color. Why? It is called homozygous recessive. It is having both the chromosome, X chromosome, that is black, so it is a recessive character, it will express. Let us think about both of them are orange and it is a dominant character. Both this animal look orange in color. Then the problem comes when you have got X and X, orange X and black X together. So if as per the Mendel's theory, this organism should appear orange in color because orange is dominant color, which should be the case. But that doesn't happen. Why? Because it is randomly inactivated. It is situated on a X chromosome. So dominant or recessive comes into picture if you have both together. It is just like you can play badminton and win a match if you have an opponent, and you are playing alone means some people play chess, both the movements you are only doing. If you are doing both, you win also, you lose also, you win, because both cannot win together, both cannot lose together in the chess match. Here what happens, dominant and recessive comes into picture if both are there in one cell. That doesn't happen. Why? It is in the X chromosome. Here what happens, some group of cells will have black color, some group of cells will have yellow in color. That is what you see in this cat. You have orange color or black color together. So it will be an intermediate size, and that has to be a female, and that female should have got, this doesn't mean that this is not a female, this is not a female, this is also a female, this is also a female, but it has got both the X chromosome having one color. Although random inactivation is happening there also, but it is not detectable, you can't detect it. But white can come, there can happen in some group where this coat color is not expressed at all, then it will appear white in color. Just like you may have seen in some humans also, we call it as epistasis or albinism, etc. Some people will have their brown skin, but some part of their body that will be milk white, absolutely white, because they are not producing melanin. So if you don't produce melanin, you will look like an albino. And some people body, some group of cells do not produce melanin. So that has nothing to do with, white is not a color. Absence of color is white. So random X inactivation in calico cat, what happens? Gene for coat color has got two alleles. XB, that is X, the capital B indicates that is for orange color coat, and then X small b meant for black color coat. Orange allele is dominant over black any given situation. Therefore, the heterozygous female cats with X capital B, X small b should have been orange in color technically, but it is not the case. Why? The heterozygous cat with X capital B, X small b shows color patches due to random inactivation of X chromosome in different cells of blastula. So why it did not show orange? Because both X chromosomes are not expressing in one cell to have an orange color. So some cells, the orange bearing X chromosome is inactive. So naturally in those cells the other X chromosome is active. So that will show naturally the black color. So that's how the calico cat is formed. Now let us see how the imprinted inactivation and random X-inactivation. Do we have, like imprinting, I already told you that imprinting is following a particular pattern with no obvious reason. It is following the same pattern as it is. It has to follow. Like you may also see in some family, grandfather was MBA and father is MBA, son also MBA. You will also see doctors. So grandfather is or grandmother is doctor, son or daughter is doctor, and their next generation also doctor, advocate. So this is, I won't call it as imprinting, but that is what the message, the instructions they get right from childhood. So they also will have an inclination to become so and so. Same way, some genes or some chromosomes get an imprinted behavior in terms of its expression. So X-inactivation can be either random or it is directed. Paternal copy is inactivated, means father's X chromosome during fertilization. Father also contribute X chromosome, mother also contribute to the X chromosome to a female in any mammals you are discussing. But some animals, one of them, paternal copy is inactive. In some of them, any one of them, paternal or maternal. So that is what, let us see how does this happen. So in a 4-cell stage embryo, the paternal X coated with exist RNA. So you know, if a chromosome, X chromosome is coated with exist RNA, outcome is it will be made inactive. And it will go until morula stage. Morula stage means a group of cells. It hasn't formed the blastula structure. I have to touch upon some of this embryology terminology. I am guessing that you have some idea about the embryology, because normally zygote is the single cell stage, then it becomes 2 cell, 4 cell, 8 cell, 16 cell, like that it goes up to around 500 to 1000 cell stage. We call it as morula, and after that it will become a blastula stage. Until blastula stage, the cells have got stem cell characteristics, means they can be called as embryonic stem cells. After that they will start differentiating, that is gastrula, where the cells start becoming 3D, that is 3-dimensional. That means you have ectoderm, endoderm and mesoderm forming, and the organogenesis and the organs are started forming and the embryo started developing. So until around morula stage, it is continuing as the paternal copy remains inactive. At the blastocyst stage, that is a stage in the blastula phase, asynchronous replication of paternal X chromosome takes place, and the macro H2A, which is the chromatin or the histone associated with the inactive X chromosome, association with the paternal X is seen until the blastocyst stage, and it will continue further for some more time. And the extra embryonic tissue, extra embryonic tissues are what? Normally amnion, allantois, chorion. So these are the main extra embryonic tissue, and in some organism it will be kind of replaced with yolk sac or some other associated structures, placenta, etc. So those who study embryology can understand this, but extra embryonic tissues can have a unique set of the embryo itself is giving rise to the extra embryonic tissue also. You should understand the fertilization takes place between sperm and the egg. Both are single cell stage. So if an embryo has to develop, the entire thing has to come from the embryo itself. So the extra embryonic tissue also derived from the zygote itself, and this eventually becomes, it will continue in the extra embryonic tissue also the paternal X-inactivation stage. Right from 4-cell stage embryo onwards, morula and blastocyst stage, until then the X chromosome of paternal, means father's copy, remains inactive, and even this continues in the extra embryonic tissues. Like in case of human, the prominent extra embryonic tissue is the placenta, that is the attaching of the embryo to the uterus wall of mother through the umbilical cord. So that uterus wall structure is called placenta, and that is very important structure for the growth of the embryo. So embryo and placenta, or embryo and the mother, is connected via the umbilical cord. So this umbilical cord derives nutrition from the placenta. So placenta and other extra embryonic structures will have paternal X chromosome remains inactive. So let us see what happens in embryonic stem cells. So X-inactivation in ES cells, unstable exist RNA expressed from paternal or maternal. So the zygote, the blastula stage, I told you that resembles the embryonic stem cell. So exist RNA coating of the paternal or maternal X is possible, means some difference. Like in the normal embryo we saw it is exclusively the paternal copy, whereas in stem cell it can be either paternal or maternal. And late replication timing happens with the exist coated X chromosome, and histone hypoacetylation takes place, and the MCB association, that is macro H2A chromatin association, and methylation of various promoters or on the genes which is present in the X chromosome takes place, and inactive paternal or maternal X in the embryonic tissues takes place. Means in embryonic stem cell it can inactivate either paternal or maternal X chromosome in a uniform manner, but whereas in an organism it is constantly maintained up to the extra embryonic tissue, the paternal copy is made inactive. So this will of course will change, like later in the blastocysts there will be a randomization event takes place where either maternal or paternal can be made inactive in the organism. That is why, like you saw in the case of cat, randomly some X is inactive, randomly some X is inactive. So this will continue in the throughout the lifespan of the organism. So we will continue more about the X inactivation in the next class. Thank you.